Good morning, everyone. We are getting ready to have our next to last virtual breakfast this morning, and I'm pleased to have Dr. Erin Hill as our specialist talking about weed seed collection. Before she starts, though, I would like to say that we do have an evaluation, and as we finish this season, I would really encourage everyone to go to the chat box and please complete the survey. We really need to utilize as much information as we can as we try to make this series better each and every week as well as each and every year. And so as we close down the season with just a couple weeks to go here, we really utilize the information that you provide on those evaluations. And I just take 5 minutes, so I'll stop talking and I'll have Erin Hill talk to us now. Erin?
Thank you, Phil. Good morning, everyone, and thanks for joining me today. We're going to talk about a little something different that you might not think about usually, and that's harvesting weed seeds. And before we get started in that, though, I wanted to point out this is the group I work with on campus. I'm in the MSU Plant and Pest Diagnostic Center. So we have a new name. We used to be Diagnostic Services. We changed it to make it more clear that we work on plants and not animals. So new name, same great service. If you don't know us already, we're a multidisciplinary team that is there to address plant health issues for the variety of sectors in Michigan. And so we have an entomologist, pathologist, nematologist, and then I am the weed scientist in the group. So before we get started talking about how to harvest weed seeds, I just need to give you a little intro into the status of herbicide resistance in Michigan thus far. So we've really been tracking resistance in Michigan beginning as early as 1980 when we had the first triazine-resistant common Lambsquarters confirmed. And we really upped that in the last, say, 10 years where we've been doing more regular screening of potentially resistant weeds in field crops. And so we've created this cumulative and interactive map on the Plant and Pest Diagnostic website, which is pestid.msu.edu. So you can go there and find this map and click on your county, and it'll tell you all of the herbicide-resistant weeds that we have confirmed in that county and what they're resistant to and when we identified that resistance. And then another way to look at the status of herbicide-resistant weeds is to visit MSUE News and check out the annual report. So every year after the screens are done, I write up a report and tell you what the latest issues are, and you can check that out there. So every year in the lab, we receive up to 70 samples for screening. It's been a little bit lower the last couple years. I believe I had just over 40 last year. And most of the samples of weed seeds that are coming in are from field crops. But we are seeing an increase in submissions from other cropping systems like Christmas trees, vegetables, and even some of our nurseries. When we're testing for resistance, usually the person submitting the sample is interested in one particular site of action. A lot of times they're interested in whether or not the specimen is glyphosate resistant. But if we have enough seed, then we actually test for resistance to multiple sites of actions to look for any cases of multiple resistance. So we usually have an ALS product in there, like Harmony. We have a synthetic auxin, which for all of our tests now we've added dicamba due to the technology that's been released. Sometimes we also look at 2,4-D. And then we look at several others like atrazine, in addition to glyphosate and glufosinate, which is in Liberty. So our newest findings, last year we did find a Palmer amaranth sample that did show some low-level PPO resistance, so that would be to fomesafen or Flexstar, and that was in Mason County, and it was also resistant to glyphosate. So other states nearby have had PPO resistance in Palmer amaranth for quite a while, but this was the first case where we had it in Michigan. We've also had the first case worldwide of clopyralid resistance in common ragweed. This came from a Christmas tree planting. So clopyralid would be the active ingredient in Stinger. And this is pretty significant just because, like I said, it's the first case worldwide. What was good, we did further testing on this and we did not find that population was what's cross-resistant to any of the other synthetic auxins like 2,4-D or dicamba. And then right now we do have some tests ongoing for a suspected case of clethodim or select max resistance in large crabgrass, but we're still at the beginning stages of that. So what is the process for screening for herbicide resistance? Well, we in our lab conduct greenhouse bioassays. So we actually are growing the plants and spraying them. And so in order to do that, we have to have mature seeds, and then we have to treat them to break dormancy. Unlike our crop seeds, which have been bred to germinate when we want them to, weed seeds don't usually behave in such a manner, so we need to use little tricks to get them to germinate. And some of those we can do within a day, and some of them it takes up to 6 weeks to do, like the ragweed species. They actually have to go out in a mesh bag and be in freeze-thaw conditions for about 6 weeks, and then they're ready to germinate. After the seeds are ready to germinate, we place them in the growth chamber in flats. And then once they've germinated, we transplant them into larger pots. After those seedlings reach about 2 inches in height or diameter, that's when we apply our herbicide treatments. We have a single boom sprayer that's attached to our greenhouse facility, and so we treat them all there and under very controlled conditions. And then 2 weeks after that treatment, we go back and do a visual rating to determine if resistance is present. Now, it's important to point out that I also have a known susceptible of each species that I treat at the same time. So that ensures that all of the process is working properly and there wasn't any kind of equipment failure or other issues. And if we suspect a new case of resistance, like I mentioned with this potential crabgrass, then we do some additional screening where we do a full-on dose response, and we may also look for cross-resistance within that site of that or that mode of action. So what we're really here to talk about today is how you could submit a sample for resistance screening, what you need to do, and what you need to look for in these seeds. So the species that we currently screen on a regular basis include Palmer amaranth, water hemp, horseweed or maresale, and the 2 ragweed species, common and giant, and then also common lambsquarters. For those of us that grow soybeans, we're very lucky to have support through the Michigan Soybean Promotion Committee through the checkoff dollars. So screening is offered for free to people who have soybeans in their rotation. If you don't have soybeans in your rotation, then there is a $90 fee per sample. And if it's something new or a different weed species, then you just need to consult with me to make sure that we actually can get that weed to grow and that we have a known susceptible so that we can do that, and that we test the products that you're the most interested in. So it's really important in order to get all of these plants to germinate and grow that we have high quality seed. So knowing what to look for in those species is important, and now is the time when we're starting to see seeds mature on the plant. So usually late August, September, and October is when you're going to find mature seed on the weeds that you think are resistant. When you do determine that you have seed that is mature, which we'll talk about in a minute, it's important that you pool samples. So we don't want just the seeds from one plant. We want the seeds from 5 or more plants in the field suspected if you have it. And it's really a numbers game. We need enough plants to survive that dormancy breaking process so that we have enough that germinate and are all the same size so we can treat about 65 plants all at the same time. So now we're going to talk about the individual species that I mentioned that we screen for regularly and tell you where to look for the seeds and what they look like on the plant. I'm going to do my best to describe this in words for those of you who are not able to see the screen this morning. But if you do want to go back and look at this later with the pictures, you can always go back and look at the recorded presentation that's archived online. So starting with horseweed or maresale, you're going to find the seeds formed at the tip of the branches. And you're going to notice that the plant is starting to produce flowers and then seeds once it starts to look very fluffy. Because similar to a dandelion, these seeds have what's called a pappus on the end, which is a fluffy structure that facilitates dispersal by wind. So once that plant starts to look fluffy, that's when you can go and start looking to see if it has any seeds that are forming. Now, unfortunately, the seeds of maresale are very tiny. They're about 1 millimeter. So if you have one, it's best to take a hand lens out there so you can actually look at the base of that pappus and see what your seed looks like. Now, early on, they're going to be almost translucent or a very light yellow color. That's the immature seed that is still forming, and that's not going to germinate in the greenhouse. What you're looking for is a seed that is slightly plump and more of a yellowish brown color. That means that it's mature and ready to germinate in the greenhouse. So that is a mare's tail. Now, I put another slide in here because this is one of the #1 species that we get submitted. And so it's really important to look for seed in this species because it's so tiny. This is the one that most often needs to be resubmitted. So I usually look at them when they come in and they've been cleaned off the plant under the scope, and I say, I don't think there's enough seed, and then I will contact you and say, I need you to go back out. So in order to reduce that time and to reduce your postage, try and look for this ahead of time so that I don't need to ask you to resubmit. The next weed species that we'll talk about is common Lambsquarters. So the seeds on common Lambsquarters are formed at the tip of branches. They're a little bit bigger than horseweed or mare's tail. They're 1.3 millimeters, and they're going to be rounded in shape and black or brown in color. So with this, you really can't look at the plant and see if the seeds are still green. You really need to take a piece off and pinch it between your two thumbnails and then look to see if you're finding any black or brown seeds. If you're finding black or brown seeds, then it's time to start collecting these plants and submitting them to the lab. The ragweed species are another set of species that are a little bit tricky because on the ragweed species, the male and female flowers are on different parts of the plant. So similar to corn, we're having the pollen formed at the top of the plant, and then the female flower, where the seed is going to be, is somewhere else on the plant. Now, it's not as obvious as a corn ear, but the seeds on the ragweed species are formed at the base of leaves and branches. So sometimes I do get people who just snip the top off and send that in. Well, it's the male flower, there's no seed there. So you need to actually submit more of the plant for the ragweed species in order to get the seed. Now, the seeds of these are even bigger than the previous two we talked about. They're about 3 to 4 millimeters for common ragweed, and my favorite weed seed to work with, giant ragweed, is relatively huge in the weed seed scale, up to 10 millimeters in size. For both of these seeds, even though they're different in size, they look similar, so they're kind of an oval shape, and at one end there are several points, and there's usually one prominent one from the very apex of the seed. To me, it kind of looks like a crown. So if you're seeing those kinds of seeds in those bases of leaves and they're a brown color or a gray color and not green anymore, then that means they're mature and ready to collect. Then the next and the final species I think that we'll talk about are the Amaranth species. So the ones that we're usually most interested in are Palmer and water hemp, though we do testing for red root and powell. Those two that are our native pigweed species, we haven't seen any glyphosate resistance in at this point in time, but we're still willing to check for that. So for all of these Amaranth species, I take that back. For Palmer and water hemp, the two that are not native to Michigan, they have male and female plants. And the seeds are only going to be found in the female plants. So again, like Lambsquarters, it's important to look for the seeds. They're located in the seed heads at the top of the plant. It looks very different from the leafy portion. And these again are small, around 1 millimeter in size. They're black to brown, similar to the common Lambsquarter species, and they're rounded and they're very shiny. So these are even shinier than the Lambsquarter species if you've seen those seeds. So again, for Palmer and water hemp, it's really important to make sure that you're collecting a female plant because the male plants I'm not going to be able to do anything with, then I'm going to contact you and ask you to go back out and get female plants. With the other two native pigweed species, if you're submitting those, which are powell and redroot, you still need to check for seed, but you're going to find seed on every plant because those ones they have both flowers on one plant. Another thing that I added in here for the Amaranth species, a lot of times people tell me that it's Palmer and it ends up being water hemp, or they think there's some misidentification of the Amaranth species because it's really difficult. Some of those native ones, it's very difficult even for us in the lab to determine the differences. So in order to help us out and make sure that we are calling it the correct weed from the get-go, for these Amaranth species, if you could actually send in some leaves that are attached to the stem, that will help us out with the ID. We can do a little bit of ID just from the seed head, but if we have leaves, that's even more helpful. And then we can differentiate that right from the beginning and determine which Amaranth species it is. So now that you have your weed seeds, you need to package them and send them or bring them into our lab. And this is a different process than for submitting other samples to our lab, say for pathogen analysis or for nematode analysis. These weed seeds need to be treated a little bit differently. Probably the biggest difference is that I would like these samples to come in sealed paper bags because when you get a plastic bag, any green material in there can get moldy. And it's not a pleasant situation for us to have to clean, and it's also not good to maintain the integrity of those seeds to have mold growing in them. So if you could use paper, that would be ideal. If you're gonna submit it by mail or even bring it in your truck to campus, it'd be important to double contain that bag. So put it in another container, like a sealed cardboard box or something like that. And really the reasoning here is that if you do have a resistant weed, we don't want to be spreading that seed across the state as it makes its way to our lab. As I said, you can bring it in person or you can mail it to our address that's listed on our website, or it can be found numerous places. Otherwise, it's also in the back of the weed guide. And then you need to include a completed submission form that notes, in particular, who's submitting it, where it came from, and then who we should be sending the results to. So after our screening process is done, we'll contact you with the results. And you have to realize that it's going to take probably 3 or 4 months after submission, depending on the species that you submit. So something like mare's tail can get treated right away or get planted right away. It's also going to depend on the volume of samples that we have. So in years that we have 70 samples, it takes us a lot longer to work through things than when we have 40 samples. And all of this time accounts for the things that I talked about already, for the seed cleaning, the dormancy breaking, and all of that growth time in the greenhouse, and then the evaluation period. There is a document that I came up with a few years ago to help you in collecting weed seeds. So this is another good resource, especially if you don't want to watch the video of this over again. It's called Tips for Collecting Weed Seeds. And this can be found on the Plant and Pest Diagnostic website. It can also be found at msuweeds.com. And here it has some of the same pictures that I showed today on where to find the seeds on the plant. It also has a nice table that shows you what the seed shape should look like, the approximate size and the color, and then the location on the plant. So this is a good resource if you're having trouble locating the seeds. And with that, I guess I have time to take questions. I did put a little reminder here on my final slide that the samples are due by mid-November because we do need to get them out to treat dormancy, especially for the ragweed species. And so we kind of have to have a cutoff time in order to get your results back in a timely manner. Thank you.
Erin, I do have one question for you, and it's
Sure.
Please quantify what is enough or the number of seeds needed to be able to do the 9 chemical tests that are shown on your slide.
All right, so that is a pretty variable answer. I don't think you could send me too many seeds is my real response. So we recommend that you collect from 5 plants. So if you have Palmer or you have mare's tail, if you can select 5 of those plants that are out there, and remember 5 female plants for the ones that have male and female plants, that usually gives us enough seed. If you send it in and there isn't enough seed, I'll contact you and say, hey, I think we're going to be limited. But usually I would say a couple hundred seeds to a couple thousand seeds, just because we need them to all germinate at the same time and be the same size at the time that we spray. And we're trying to get 65. but it does take several hundred seeds in order to do that. The other thing I'll say is if you suspect that it has a new resistance issue that hasn't been previously identified in Michigan, I need even more seed because I'm going to run this test multiple times. And if I need to run the dose response test to really quantify the level of resistance, that requires even more seed. So some of these specialized cases, you know, I would ask for probably as much seed as I can get. Because if I run out of seed, which is what happened with this, this crabgrass, and why I don't really have an answer to that yet, I actually have to grow the seed from the seedlings in the greenhouse. So that takes the lifetime of a weed to produce and collect. So you can't send me enough is my real answer.
All right, Erin, I think we're out of time this morning, but I want to thank you for an excellent presentation.